I watched a little more of Are You the One? They got their first match. They got 1 week left. I can't even go into it. It's too much. Okay. So I think this is where we left off. So we talked about the saccule and the utricle. We talked about static equilibrium and linear acceleration. Now this is the dynamic equilibrium. This is movement in different planes. And the things that do that are basically the semicircular canals. And the structure that's doing this is the crista. So if we look at our inner ear, this was the cochlea for hearing, this was the vestibule, okay, this was more of the static equilibrium, and here's the dynamic equilibrium with the semicircular canals. Each one of these canals is at a different angle. Okay, there's one at like 0 degrees, 90 degrees, and like 30 degrees. And it's real simple how this works. The crista, there's one crista in each one of these tubes. It's filled with fluid. It's endolymph. So depending on how you're moving, it may cause the fluid to move through the tube, push on the crista, and bend the hair cells. Okay, that would be the stimulus that would get picked up on. And I want you to imagine if these were hula hoops. I don't know if you guys are too young to know what a damn hula hoop is. You guys know what hula hoops are? One of those at like 0 degrees, okay? And somebody was spinning in that direction. In fact, they used to have little things in there that made noise that spun, and they were moving in that would stimulate that hula hoop. But if you had another one at 90 degrees, spinning in that direction wouldn't do anything, okay? Spinning in that direction with one at 30 degrees might have a little bit of stimulation. So your brain gets all three of those inputs and can tell what direction you're moving, what's going on. Does that make sense? This one that's like this, if you were doing more of a head over heels somersault type thing, it would stimulate that. The fluid would move through there, push on that crista, but the one at 0 wouldn't be getting anything, okay? And it still also tries to kind of put that same information with what your eyes are telling it, processing that whole thing. Does that make sense on how that works? No, it's more dim. It's because you got these in both ears, so fluid will bend the hair cells. So it doesn't matter which way it's bending the hair cells. So your body kind of has to process that still with vision and whatnot on which way you're spinning or what. Okay, think of and this still happens like when you do like a merry-go-round, like your little kids and you spin people, or you take a drunk person and you spin them around and they don't know, like they're basically going to fall over. You're messing up those signals that they're getting, and they're still feeling that perception of that spinning because of this stimulation to these hair cells. So hair cells are buried in this gelatinous membrane. We have one of these per canal, and these are our cupulas. Okay, so this whole thing is the crista. The gelatinous membrane itself is called the cupula. Duct orientation causes different ducts to be stimulated by rotation in different planes. So that's what I was trying to explain with a 0 degrees and a 90 degrees. Spinning in one direction will affect one and not the other, okay? And then that 30 degrees might get some minimal stimulation, might not. Your brain processes all that information, okay? So here we have our gymnast standing there, not moving. The fluid, the endolymph, isn't being pushed either direction. We don't bend the hair cells. We don't get stimulation. Here she does this, which might be called a pirouette. I'm just guessing anymore. Fluid moves through there, pushes the cupula where the hair cells are, bends them. That opens them up, and we get depolarization, stimulation on that vestibular nerve. So endolymph pushes the cupula and stimulates its hair cells. Okay, so both of those dynamic and static equilibrium inside the inner ear, it's a very simple mechanism. Okay, equilibrium pathways, hair synapse on the vestibular nerve, we know that's going to come together with the cochlear nerve, that's our cranial nerve 8. Fibers in the cerebellum, okay, which makes sense. We know the cerebellum has a lot to do with coordination, balance, depth perception. And nuclei of cranial nerves controlling the eye, head, and neck. So that's like cranial nerve 9, cranial nerves 3, 4, and 6. And why would we send signals there? Okay. Well, equilibrium, basically, is kind of where you are in space. So depending on what's going on, we want to send information as quickly as we can to your eyes and head so that you can get if you got knocked over, okay? You're a UFC fighter and you just got punched and you got dropped and you fell down. You want the signals getting there as quickly as possible so you can get your eyes up on that person coming in there to finish you off type of thing so you can protect yourself, that type of stuff. That takes us to vision and light. Okay, for vision, what is the stimulus for vision? What's stimulating the retina, the rods and cones? Light. Okay, so it's all about light waves, and it's light waves being reflected off of objects. Okay, you guys probably knew that. So when you see the color red, that's light waves hitting whatever that is, and the red wavelength is being reflected back at you, hitting your retina, stimulating your cone cells to perceive the color red. If you perceive the color white, then all the light rays in that all the wavelengths in that spectrum are being reflected back. And if the color is black, then none of the wavelengths are being reflected back. That's why they have you wear light colors in the sun. It does give you a little bit of a heat reflection, not enough to do much good out here, but a little bit. So visible light's electromagnetic radiation. The wavelengths are between 400 and 750 nanometers that our eye can perceive. Now, there's ultraviolet light, there's infrared, there's wavelengths out there that we just can't see. Some of them, animals have a little different wavelengths, so they can see things some of those things a little better than we can, or things that we can't even see, like maybe ghosts. How many people believe in ghosts? There we go. There's always some people, I don't know. How many people believe in aliens? Well, there we go, see? So, anyway. The external eye, you guys probably know all the features of the eye. We got the eyebrow, got the eyelids, eyelashes. Okay, so eyebrow, eyelids, eyelashes, none of that's new. The conjunctiva, now that might be something. That's a mucous membrane, a clear mucous membrane. So transparent's the word they're using. That is underneath the eyelid, both the upper and the lower, and covers the anterior surface of the eyeball, except for where the cornea is. So over here on this picture, if you could grab his lower lid, he would have a membrane that goes underneath his lid and comes back up onto the eye to about this point, okay? And because it forms this kind of U shape, it's what keeps anything from getting down, back, around behind the eye. Same thing up here. If we could pull his upper lid out, there's a membrane that lines right up underneath it and then comes back down on the front of the eye, okay? That's the conjunctiva. It's richly innervated, it's highly vascular. Anything that has a good blood supply typically heals fairly quickly. And so the next thing we're going to do is, I kind of mentioned feels like last week, but this past Tuesday, that vision is kind of a lot of these special senses are learned. To hear a noise, to be a baby and hear a siren is one thing. To know what that siren is that a fire engine? Is that an ambulance? Is that a police car? Or that type of thing. You learn what these sounds are. You learn what these things that you see in the world are. So what we're going to do is I'm going to put pictures up. I'm going to put them up for about 3 seconds. So I'm going to put it up and go 1001, 1002, 1003. So what I want you to do, you got those little lines, in your own little shorthand. Like, don't write out a paragraph, just something. Look at it at 1001 and then jot down what you saw. Okay? And I'm going to show you why, true eyewitness accounts of things aren't really good. Okay? It's why if you go into like a 7-Eleven or a Quick Stop, when you walk in, have you ever noticed at the door they have the lines for basically height? Okay? That's so they can get an accurate estimation on the height of somebody that comes in and robs that place. Okay? Because if they don't, and there was 3 people there, somebody's going to say they're tall, Two people are going to say they're short. Their descriptions of them are going to be all over the board. Okay, they train police officers and whatnot to look for certain details and specific things, and a lot of what we see was learned. It has to do with our past experiences. All those things shape us. That was kind of fun. But notice that we all see different things. Some of us saw things maybe in more detail, others not. And that's just pointing out that these special senses, the sense of smell, the sense of sight, the sense of hearing, it's all learned. Like you have the ability to like smell something. And you ladies would be much better at than me. I might be able to smell something and not be able to put a name or a description to it. Where you guys would be, oh, that's definitely this. Colors are the same way. Men are pretty simplistic with colors. Like, oh, that's blue, and that's red, and that's green. And they're like, oh, no, that's magenta. That's whatever, sunset orange, whatever. And you're like, oh. It's like you guys got that big box of Crayolas. We only got that little 6-pack. All right, so structures of the eye. So this is kind of a weird looking picture. What we have is we have our eyelid here in front, our eyelid here on the bottom, our eyelashes. Okay, here's that clear windows of the cornea. Here's that conjunctiva underneath the eyelid and then on the anterior surface of the eyeball except where the cornea is. Behind it, this is the iris. The iris, this is what changes shape to make the pupil bigger or smaller. The pupil was just the space. The lens is what was behind it. It was really hard on those cow eyes. And here's that ciliary body here and here, and it's got ligaments that attach to the lens and can change the shape of the lens a little bit to kind of fine-tune the image. Okay, so that's kind of what that picture's trying to show us. Now here, on this picture, we're looking at the right eye, and both eyes have what are called lacrimal glands, and the lacrimal glands are in the superior lateral aspect of the eyes. All right? And so the lacrimal glands drain through the lacrimal ducts. The tears wash across the eye from superior to inferior, from lateral to medial. They get taken into the lacrimal canals. Well, actually, they start what's called the lacrimal punctum, to the lacrimal canal, into the lacrimal sac, and then down the nasolacrimal duct to the nose. That's why when you cry, your nose will run, because the tears are actually washing through down your nose. Okay? So lacrimal gland, we had lacrimal ducts, lacrimal punctum, lacrimal canal, lacrimal sac, nasolacrimal duct. That's how I feel too. What's that? Possibly. All right, so extrinsic eye muscles. We've mentioned these. We've even talked about which ones the cranial nerves control. Okay? So they're the muscles that insert onto the eyeball. Okay? And we had 6 of them, right? We have let's say this is a right eye. This is a medial rectus. It contracts, it's going to pull the eye to the medial. We have a lateral rectus. It contracts, it's going to pull an eye lateral. Superior rectus is going to pull it up, and inferior rectus is going to pull it down. Okay, so those are the 4 rectus muscles. Then we have a superior and inferior oblique. The superior oblique pulls the eye down and out, while the inferior oblique pulls it up and in. Okay. Most of these were innervated by which cranial nerve? 3. Okay, good. There was one that was done by the trochlear and one that was done by the abducens, right? Lateral rectus was done by the abducens. Trochlear did the superior oblique. Okay? So if on lab or here, where if they ever get specific on that, just remember cranial nerve 3 does all the other movements, plus it closes the eye. Does it do anything else? Yeah, it constricts the pupil, okay? So then you really only have to remember abducens does lateral rectus, the eye moving laterally. Trochlear does superior oblique. which does the eye moving down and out. Okay? So cranial nerves 3, 4, and 6 are what innervates all those. We already knew that. Yeah. Down and out is the superior oblique.
So it's above the eye.
It's kind of on a pulley system. Here they're trying to draw a picture of it. It kind of wraps through a pulley, so when it contracts, it actually pulls at a different angle than where it's at. That's why. And the inferior does the same way. It's really hard for them to draw it and show you properly, but this is the best. Like, oh, so it's kind of not running straight. Tunics of the eye. Tunics, I think we've come across that term, maybe not, but that basically means layers. Okay? There's what they call 3 tunics or 3 main layers of the eye. We have the fibrous layer, tunica fibrosa. That's the outermost layer. So what was the outermost layer in the front of the eye? What was that structure? Cornea, good. And then the white all around everywhere else was the sclera. Okay, so that's what's in that layer. Okay, then there's a vascular layer, meaning a good blood supply, tunica vasculosa. And the structures you find in there were the choroid, the ciliary body, and the iris. And then the third layer, tunica interna. This is where you're going to find it was that brown stuff that kind of gets messed up, that lays across the back of the eye. What was that? Kind of looked nasty, really. Retina, okay? Retina was one of the things, and the optic nerve exiting from, and where we knew the optic nerve exit the inside of the eye was called the optic disc, okay? Just trying to make sure you guys are all ready for your practical. So, I don't know if this is the best picture to look at those things. Here, this white and cornea would be the fibrous layer. Inside here with the ciliary body, iris, and this red here being the choroid. That's the vascular layer. The internal layer, here's the retina. And here at the optic disc where the optic nerve is exiting the eye. Here's just trying to show you those 3 layers with 3 different colors. The sclera and cornea, okay? It's the fibrous layer, the iris, ciliary body, choroid, the vascular layer, and then tunica interna would be the retina and the optic disc. The fluid in here that you don't have to identify on the test because it dries up is called the vitreous humor. Good. Optical components. The optical components are all transparent. Light has to be able to pass through them, okay? And then when the light passes through them, what they tend to do is bend, or the better word is refract, the light rays, and they try to focus it onto a certain target at the back of the eye on the retina. Okay? So we're going to have a spot on the retina that's going to be more sensitive, and if we can focus the image there, our vision is really good. If when we bend these light rays, they focus too early or too late, then we're going to have a problem with our vision. Our vision isn't going to be as good as it could be, and then you're going to have some sort of corrective lenses to bend the light, either help you bend the light one way or bend it the other way because you're focusing it too quickly. So we'll get to that in a second. So optical components conclude the cornea. We know what the cornea is, we know where it's at. The cornea is actually what refracts or bends most of the light, okay? You kind of come into this, and if you're like me, you think, oh, it's probably the lens. The lens only bends it a little bit, okay? So the lens more or less fine-tunes or sharpens up the image. The cornea is what bends most of the light, and that's how you can do like LASIK procedures. They reshape your cornea and have the light focus where it needs to be, and your vision goes back to 20/20 or even better. There's an aqueous humor. So we had a vitreous humor, and that's why I asked you a second ago, because I want you to realize there's an aqueous humor. So the divider is the lens. Behind the lens, between the lens and the retina, is the vitreous humor. In front of the lens, between the lens and the cornea, there's aqueous humor. Okay? Aqueous humor is not nearly as thick as the vitreous humor. It's more fluid, it's more serous. Okay? Light passes through that, so they consider that one of the optical components. It then goes through the lens. The lens were held by ligaments that attach to the ciliary body. The ciliary body contracts or relaxes, changing the shape of the lens. And then behind the lens was the vitreous humor, which helped basically hold that retina flat against the back of the eye. Okay. So those are the optical components. Anything that light passes through and gets bent. The neural components are the retina and the optic nerve, okay, the things on that tunica interna. And at the back of the retina, we have the target where we're trying to bend the light rays, and it's called the fovea centralis. Okay, it's about a 3 millimeter patch in the back of the eye. Okay, and this is where, if the image gets focused, we get the best picture. It has lots and lots and lots of cone cells. Cone cells are responsible for color vision and sharp contrast. Okay? It doesn't mean that if we hit other parts of the retina that we can't see it, it just won't see it as clearly. So think about receptors, tactile receptors in your fingers. Okay? You have lots of receptors in your hands and your feet and your face. And so you can discriminate between one thing touching you and two things touching you. Okay? You can do that on your fingers. Your back isn't that same way. If you take a two-pronged thing that's maybe 1.5 inches apart and touch somebody on their back, they sometimes can't tell there's two prongs because there's not as many receptors. They're all spread out. They don't get as much information. Same thing here with this fovea. The more cones we have, the sharper the detail. So it's kind of like seeing something in high definition versus seeing something in whatever was before high definition. Standard. There we go. Thank you. And the center of this is called the macula. Anyway, this is where we have lots of cone cells. This is where we get the best image picture. Okay? And those of you that had lab, that little, they didn't even have it out, did they? Oh, there was, because I don't remember demonstrating it. Anyway, they normally on the lab, they have a device. You hold it up to your head and it has an arm. And it has a little thing that you focus on right here. And it brings, this arm pivots around and brings something into your peripheral field of vision. Okay? So if you look straight ahead, it's in your peripheral vision. Well, those things that come in, they get focused on the retina, but they don't get focused in the center. And so they'll have two little letters on it. And they'll bring it around, bring it around, bring it around, bring it around. And you can't read it. You can't tell exactly what those letters are until it's within 10 degrees of being straight ahead. Okay? We don't, they took it off for some reason. Maybe it got torn up. But it's interesting because you wouldn't believe that. You'd think, oh, I'm sure out here I could tell what it is. You really can't. Okay? Well, there's a blind spot where, yeah, you can't see. But this is kind of that same thing. Your peripheral vision you can see, but you can't see details. Okay? And like the best way I could tell you is, you know, like if you wanted to mess around with that, is if you probably wrote, a couple letters on your hand like T and P, and you had somebody look straight ahead, and you put it over here in their field of vision, they couldn't read it. They could see it. They could see your hand. They might even be able to see, oh, there's some letters on your hand, but I can't read them. Now, if they do, what they did is they probably did a quick glance, because that's what you want to do. Like, you just can't help it. You're like, oh, there it is. Okay? But that's what was supposed to be on the lab to demonstrate this, that if you don't hit, if images don't hit the center, you don't see them very well. Okay? Now, when the image hits the optic disc doesn't have any retina there. That's where the optic nerve exits. So there's no receptor cells. There's no rods or cones. So there's no place to, it's like focusing a picture on film and you have a spot where you don't have film. Well, you're not going to have an image then, right? And so that's when you do the blind spot thing, that there is a blind spot in your field of vision, okay, that your other eye usually covers for you. Or if you only have one eye, you block the other, you have a hole there, but you don't see a hole. Your brain tries to fill it in with what it thinks is happening there. Okay? And the whole brain fills it in with what it thinks goes on. That's a little bit of sometimes what magicians use to try to trick you, or things to scale. And I'm trying to think of how, where they can shoot, like on TV, they can shoot something in a small version, and you convince yourself that it's lifelike, okay? But it's really on a small scale. Anyway, it's your brain. They can trick your brain into thinking what's going on. So this is where the optic nerve exits. There are no receptor cells. If you've ever wondered what it looks like when they look in your eye with the ophthalmoscope, this is what it looks like. This is just a drawing. This is actually what it looks like. They can see the optic disc. Not only did the retina or the optic nerve come in here and innervate the whole retina, it's also where the blood vessels come in. Okay? And then here in the center is this fovea centralis. This is where we're trying to hit things with the image, but not everything hits there. Okay? Because we only have so much ability to bend the light. Okay? There's another thing that's not on our test. If you have kind of, say, 20/40 vision, what's happening is the light rays are coming in, and some of them aren't being bent enough to hit your eye, and it distorts the image. It makes it a little bit fuzzy. So if you take a piece of paper and you punch like a very small pinhole in it and you look through it, that blocks all the light rays that need to be bent, and the only ones that are coming in at about a 90-degree angle. So they go straight back. Okay, if something hits something at 90 degrees, there's no bending it, right? It just continues to go on straight. And if you do that, you can actually see a little better. So what you see is you see the blood vessels running through here. Sometimes you can see if somebody has like glaucoma, what's happening is the vitreous humor, the pressure is building up in here, and it can actually restrict the blood flow, which is going to damage the retina. Some people with hypertension, you can see it looks like little pits that's damaged because the blood's coming in there at too high a pressure. Okay. Formation of an image. So first of all, the light must pass through the lens, and it gets flipped upside down and backwards on the retina. Okay. The other thing the eye does is we have the pupillary constrictor and dilator. And we think of that a lot of times for how much light we want to let in. Okay? If there's too much light, that blinds us. If there's not enough light, we can't see. All right? The other thing it does is what I was just talking about with the pinhole. If it constricts, that constriction blocks out some of the rays of light. So imagine a kind of a convex or concave surface here. That's your eyeball. Okay? And anything coming straight in will go straight back and we'll hit that fovea. As we get away from that, even if it's coming straight, it has to get bent. Okay? And what happens is, if this isn't shaped well and we don't hit our target, let's say X is our target, if we constrict the eye, we limit some of these light rays out here that are making the image distorted. Okay, so when you start squinting, you're trying to see something and you're squinting, you're trying to limit, you're trying to shrink this down so that maybe you're blocking this, and the light rays that you're getting are just from here. Okay, does that make sense? Okay. Good, because I didn't have a plan B on explaining that. So pupillary constrictor, smooth muscle cells that encircle the pupil. Pupillary constriction, was that parasympathetic or sympathetic? Parasympathetic, good job. All right, so that's what narrows the pupil. The pupillary dilator are spoke-like arrangement of myoepithelial cells that are under sympathetic stimulation that will widen the pupil. Okay, so pupillary constriction and dilation, two different muscles, two different innervations, but a kind of a unified contrasting effect. So here's an eye. The pupillary constrictor muscles wrap around it like this, like they're going around it in a circle. And when they constrict and they bulge up, it does this. Okay? And it narrows down the pupil. The pupillary dilator muscles are more like spokes on a tire. When they contract, they pull this direction. And so they pull everything this way, and then you go from this, to the dilated thing. And you guys will see that when you shine the pin lights in people's eyes. And when you're maybe smoking weed or whatever you're doing, and the police officer shining a light in your eyes, that's kind of what he's looking for, to see that there's this reaction. Because if they're not, if they basically just stay like little pinholes, you're in that rest and digest. He's thinking something's going on. Not to mention it usually reeks pretty bad, so they're usually on to you anyway. All right, so refraction, we've already touched on what that is. Bending of the light rays occurs when any type of light rays hits one of those optical components at any other angle other than 90 degrees. Most of the refraction was the cornea. The next bit of refraction happens at the lens, but it's a much smaller amount. Okay? And this will have to happen when you shift your gauge between near and distant objects. Okay? The advantage, when you get older, I'm even getting there, God, it's horrible. Things off in the distance, all the light rays are generally coming in at kind of a straight angle, kind of what I drew here. They're coming in straight, but they have to be bent, okay? If you have an object up close to you, you know, mom or dad, grandma, grandpa, my old butt, when you put a book in front of us, now the light rays are actually coming in at angles like this. So not just straight, they're coming in like this, which means they have to be bent a lot, okay? And we lose that ability to shape the lens and bend the light rays that much, and that's why they can't see things up close. You ever hand mom something to read and she's like, the hell does that say? Okay? I've started doing that. It's embarrassing as hell. Okay? That's what's happening, is they've lost some of the ability to change that shape of the lens. That ciliary body isn't as strong, the lens is not as pliable, and they can't do it. Okay? Or you'll start to know you're getting to that point when your eyes fatigue reading. Okay? Because you're doing that when you're reading. When you're young, it doesn't even matter. You get bored reading, but your eyes don't get tired. You get older, at a certain point, you're kind of like, God, it just makes my head hurt. I'm getting tired. That's because those muscles are, they're just not used to it. They can't do it as long anymore. Okay? So near response, when we have distance vision, we receive most of the light rays running parallel. That's what I was trying to say. Something off in the distance, they're mostly coming parallel straight to the eye. We still have to bend them, but they're coming in at some angle 90 degrees, at an angle of 90 degrees, and then have to be bent, okay? And we can focus this without much effort, okay? Near vision, when we focus on something up close, now the light rays, now the light rays might be coming in like this. Okay? And see how some of these are coming in at something greater than 90 degrees. So to hit here and bend it back to hit this spot requires lots of effort. So that's why their eyes would fatigue out, or if they can't do it, why they can't see those things. It also requires convergence of the eyes. So your eyes try to move in by turning your eye in, is trying to capture that angle and take it more to a 90 degrees, okay? So that fatigues their eyes out. We'll constrict the pupil when we do this. We already talked about why we do that. That takes some of the light waves on the perimeter out of play, some of the ones that need to be bent the most. We'll do some accommodation with the lens so that the lens is bending the light even more. Okay? So the lens, think of the lens as either being flat, okay, with things kind of passing more or less straight through it, or it bulges, that now is bending the light more. When we let that lens bulge, it requires constriction of the ciliary body and bend the light, but that's, once again, some sort of muscle contraction in the eyes that will fatigue out. So here we're changing the length of the eyes. When this ciliary body bulges, The strange thing is we're used to muscles pulling something. This ciliary body bulges, it actually puts slack in these ligaments, and this lens is allowed to thicken up and bulge, which now it's more rounded, it's going to bend the light rays more. When the ciliary body relaxes, it falls back this direction, falls back this direction, and that actually pulls the ligaments tight and flattens out the lens. So the muscles relax, but the tension on the lens is changed. So think of things we're wanting them to focus back here. Sometimes light rays are going to come in here and focus too short, okay? Or they're going to not get focused fast enough, and their sweet spot would have been back on the outside of the eye. So what they'll do is they'll put some sort of lens out here to either help focus them so the ones back here are the focus brought forward, or actually if the ones focus too early, then they spread it out so that it makes it back there. I think we might have a picture coming up. Here's the distance vision. Here's how it's talking about these light rays coming in parallel. Near vision, these would be coming in at a greater angle, so we try to converge the eyes so that now they're still coming in straight as much as possible. Okay? Here, parallel light waves, they don't need to be bent as much. Here are light waves that are greater than parallel. They're coming out at an angle, so now we have to bend them even more to hit this sweet spot. Okay? Does that all make sense to everyone? Okay. When we have problems, the light rays are focused too soon. Okay? So it comes in here, we bend them, but it bend them too much. Okay? So in this person, they're nearsighted, or they got myopia. And so what happens is they give them some sort of lens that actually takes the light rays and bends it outward, so that now when it bends it, it won't focus until it does hit back here in the sweet spot. So whether they're putting a contact in their eye or glasses, that's what that lens is doing. Here's somebody that's farsighted or has hyperopia. The image is being focused behind the sweet spot, past where the retina is. So they put some sort of lens in there that starts bending the light rays before it even hits the cornea. Instead of spreading them out, it's already helping them bend them so that we can get them bent in time. Okay? All right, so the back of the retina is dark pigments, okay? And in the sheep eye, we saw the choroid. It was black. Well, the very back layer of the retina is black as well. It's dark, okay? And that's so we don't have light being reflected. When the light comes in the eye and hits the retina, we want it to be absorbed. We don't want it reflected and bouncing around in the eye. That would give us shadows. So most of us are in the digital age of pictures, but back in the day where they actually had to take film and go in a dark room, if you had too much light, it would cast shadows or reflections on the pictures, and that wouldn't be good. Okay? So it absorbs the stray light and prevents these reflections. We mentioned the photoreceptor cells. What were the two types of photoreceptors? The rods and cones. Cones are for color vision. Rods are for black and white. Okay? So rod cells are night vision. I don't care that it's photopigments rhodopsin. I don't care what it looks like. They named it based on it looks like a rod. Okay? Rod cells, the advantage you have with rod cells over cone cells give you the best detailed image, but they have to have a certain amount of light. Rods don't give you very sharp vision, but they don't need nearly as much light. Okay? So they can see in the dark. But your image of what you see in the dark versus what you see in the light, it's much more like a picture. It's grainy. You can't quite see it. It's like it doesn't have the same number of pixels. Okay? So your determination of what you're seeing isn't as sharp. Cone cells, color vision, but they need bright light. They've got kind of a cone head. That's how they named them. So here's a diagram of kind of how this looks. This retina, now we've seen it, it's very, very thin. They're giving it some thickness here. So here's light passing through the vitreous humor. It actually passes past these cells and off the back of the retina, this dark layer, so we don't bounce the light around. And here's where we have the cone cells and the rod cells, and the light hitting them stimulates them. Okay? They're going to synapse on a type of cell here in front. These are going to be bipolar cells, and then these cells are going to synapse on what are called ganglion cells. The ganglion cells, their axons are going to be bundled together, and that's going to make up our optic nerve. Okay? And it's going to go this direction and out the optic disc, and back the optic nerve, back to the optic chiasm and the optic tracts, and then it all gets projected back to which lobe for vision? Occipital lobe. Good. Okay. Here's an actual slide of what that looks like. So here's the vitreous body, the ganglion cells, the bipolar cells, the rods and the cones, and our pigmented epithelial layer of the retina. Behind that's still a dark layer of the choroid, and behind that was the white layer that was the sclera. Okay, so the receptors are the rods and the cones. The first order neurons are the bipolar cells, okay? So they're the first order neurons. They're going to synapse on the ganglion cells, and we have a large amount of convergence. So we have a large number of bipolar cells that synapse on a fewer number of ganglion cells, okay? That gives those ganglion cells the ability to process what's coming in, right? It's like votes. Yeah. Yes. The ganglion cells will then be our second order neurons or our postsynaptic neurons. And their axons come together to form the optic nerve, and we have even more convergence, okay, that takes place. And in between these, we've got two types of cells. We've got what are called horizontal and amacrine. These types of cells, the horizontal cells kind of go from one bipolar cell to another. And there's some facilitation and some inhibition that takes place. The amacrine cells are between the two ganglion cells at their synapses. And then once again, there's inhibition, facilitation that takes place. They're thought, the horizontal and amacrine, to help sharpen the image, to block out things maybe we don't need, increase things that we do need to help us see better. So they form connections between the bipolar cells and the ganglion cells, and they enhance our contrast. They make the image picture sharper. Here, we got a picture coming up to kind of show you. So these horizontal cells will be between the rods and the cones and the bipolar cells. They'll kind of be in those synapses. The amacrine cells will be in the synapses between the bipolar and the ganglion cells. So here's a cone, here's a rod, here we have some convergence onto a bipolar cell, and in between them we have these amacrine cells that are monkeying with, let's just say, the synapse, making it, facilitating it, making it more likely to fire, or sometimes inhibiting it, okay? Then we have the bipolar cells. They're going to converge on the ganglion cells. They didn't draw this real well. And in between there, we have the amacrine cells in their synapses that might be facilitating, might be inhibiting the different firings there at those synapses. Visual pigments. So some of this stuff we don't need to know. We want to know the visual pigment of the rod is rhodopsin, okay? I don't care that you know it's retinol and opsin. The name, that just doesn't matter. Some of this stuff I should take off. Photopsin is the visual pigment of the cones. I do want you to know that. And it has three kinds of different cones. Each cone, each type, has a little bit of a different color, okay? And so if you've ever mixed colors to make different colors, you know, you mixed red with yellow to get orange, or blue and yellow to get green, that's kind of how that is, okay? There's three different colors. Depending on how they fire and how they interact will give you all the other colors that we're able to see in our spectrum. Okay. This slide's important. It's showing you how we flip the image. Okay. How things. So let's say this is a right eye, this is a left eye. They're actually doing two different things, but something over here in this field of vision. So I'm looking straight ahead, something over here to my right. In my right eye, it'll get focused over here on the medial aspect. On my left eye, it would come in and get focused over here on the lateral aspect. And this is going to be important in just a second. Something in the left field of vision on my left eye gets focused medial. If this same dot was being seen by this eye, it would come in and get sent over here to the lateral. Okay? They're flipping this telephone pole in this picture. Don't care about that. Because what happens is our field of vision. So if I have something on my, okay, I thought you were raising your hand. Let's say, you know you're in trouble anytime I start drawing things. If I have something over here, okay, and the eyes are looking forward, this comes in and gets focused here to the medial. It comes in here and gets focused to the lateral, right? Okay. When the optic nerve comes out of here, there was this little X that it made, right? What was that called? Optic chiasm, okay? So this is what's called, this optic chiasm is called a hemidecussation. Hemi means half, decussation means crossing. So it means half of the information gets flipped, okay? So the way that works is anything here on the medial comes in and gets flipped, okay? If it's something over here on the lateral, it comes in, it doesn't get flipped, okay? So keep in mind, this was on the lateral, got focused to the medial, so it gets carried over here to the left side. This was over in the lateral, so where does it go? Straight back. So you see, did I just change the picture? See how they both, something in this right field of vision, how it got carried totally on the left, what would this portion be called? The left optic tract. Okay? What if somebody had, or let's say, so obviously if it was something in this right field of vision, it would get over here. What about something in the middle of their field of vision? So that would come in here, it would get focused lateral there, it would get focused lateral there, right? Yep. So they both go straight back. Where would the problem be if I told you a patient came in and they complained of tunnel vision? They couldn't see anything in the periphery. In the chiasm, okay? Because the stuff in the center got pushed to the sides and went straight back. The other stuff in the periphery got sent to the middle, and that's what crossed. So a lesion right here would give you tunnel vision, right? If somebody could not see in their right field of vision, where would the problem be? Not their right eye, their right field of vision. Like, your hand, you're doing your fingers from like, hey, I can see two fingers. Yeah, I can see two. Oh, wait, I can't see shit. Okay? Where would that problem be? Be the left optic tract. So I said right. So if this was the X, the problem would be right there, right? If they couldn't see out of their right eye, where would that be? Optic nerve. Okay? You get that, or did I just like, okay? So that's why I'm showing you that. There might be a question. There won't be more than one, probably, a question, but there could be a question like that. Either saying, what would happen if we damage this, or this is what the patient had, where would you expect the problem to be? Okay? One of those, like, thinking questions. Now, I walked you through it more than sometimes I do, but because I didn't want you to yell at me at the end of the year. I feel like if you stuck it out this far. you deserve some more help.
That diagram.
I don't think you need to be able to draw it, but you want to understand it, work it in your mind. Everybody all right with that? You kind of get what's happening? Okay. All right, just Aaron? 'Cause I'm not going to just help Aaron. I'm just kidding. Okay, so you have to look to where it gets focused in the eye. Okay? Anything in the medial aspect of the eye will cross. Okay? Anything in the lateral aspect of the retina won't cross. It'll go straight back. So you have to realize out here, yeah, so the green. See how the green went straight back? Okay? The red got sent to the other side. Okay? So something here focused here would get sent to the other side. So if we had a image over here in green, it would get focused there, and it would be taken back and crossed over here.
So medial with the lateral, and then lateral.
Mm-hmm. Yep. Yep. Okay. And then it's just as simple as knowing these two areas cross, these two areas out here don't cross. Okay.
And what were you saying about, yeah, like if something doesn't work, how do we know?
You have to think through that.
So like, what was the optic chiasm?
Well, tell me. If we had damage to the optic chiasm, what would the patient present with? Where would the problem be? What wouldn't they be able to see?
The medial sensitivity. It would be a problem.
It'd be something that got focused to the medial, right? Which things that got focused to the medial were actually from in the real world, they were on the periphery. So they would have tunnel vision. They wouldn't have good